Para-aminobenzoic acid, also known as PABA, is a benzene ring with carboxyl and amino groups in opposite sides. It is used in the production of sunscreens, dietary supplements, or even as a building block for other pharmaceutical products. PABA, produced naturally, comes from the shikimic acid pathway. This pathway is also involved in the synthesis of some amino acids such as phenylalanine, tryptophan, or tyrosine. The shikimic acid pathway is found in yeasts, other fungi, bacteria, or plants, not in mammals. But how does shikimic acid turn into PABA? First, shikimic acid gets phosphorylated by the action of a kinase. Then, phosphoryl pyruvate forms this intermediate. After phosphate elimination, we get corrosive acid. Then, glutamine transfers its amino group into the corrosive acid and forms this other intermediate as long as glutamic acid. At last, pyruvic acid elimination forms our final para-aminobenzoic acid. So, why do bacteria need PABA? PABA is involved in the synthesis of folate, and thus in the production of DNA. That's how drugs like sulfamethoxazole, sulfasalazine, or sulfadiazine work. These drugs are also known as sulfa drugs, and they deliver sulfanilamide instead of PABA, so they interrupt the folate synthesis. By chemical synthesis, PABA is produced by the oxidation of para-nitrotoluene, and then the reduction of para-nitrobenzoic acid gives the respective PABA. Other route is the aminolysis of the monomethyl ester of terephthalic acid. Hofmann rearrangement is done on the resulting amide. As I said, para-aminobenzoic acid is a very useful building block for organic synthesis. Benzocaine or procaine could be synthesized from PABA. Para-hydroxybenzoic acid, the precursor of parabens, is also synthesized from PABA. And upon decarboxylation, PABA gives aniline, which is even more useful. This video will be divided into parts. The first one is the extraction of PABA, and the second one the decarboxylation into aniline. For this experiment, you will need methanol and para-aminobenzoic acid. I used 80 capsules of 500 mg each, or 40 grams of PABA in other words. First, open the capsules and place everything inside a large round-bottom flask. Then, pour 250 ml of methanol. Start mixing with a stir bar vigorously. After approximately 30 minutes, the solution looked like this. The gelatin of the capsules almost disappeared. I tried to filter the solution, but the gelatin made it impossible, so I just decanted it off and left behind the solid residue. I added water to the residue in order to crush out all the para-aminobenzoic acid left. Then, I removed the methanol by fractional distillation. After a while, all the methanol has been removed. Then I added some water in order to break down the solid. I filtered the suspension and also filtered the PABA present in the previous residue. Then I transfer all the solid into a beaker. After that, I added approximately 200 ml of 96% ethyl alcohol in order to perform the recrystallization. When everything has dissolved, I started adding water until the stearic acid crashes out. That's because the capsules also contain stearic acid, and it is extracted along with para-aminobenzoic acid. I took off the beaker from the hot plate and manually removed all the stearic acid that started crystallizing on the surface. I dissolved the stearic acid in acetone, as you can see it is quite soluble. I left the solution in a fridge for something like 2 or 3 hours, and when I returned, all this solid recrystallized. I filtered the crystals, and unfortunately, all the stearic acid left stayed on the surface of the crystals, so I manually removed them using a spoon. As you can see, the stearic acid easily dissolves in acetone. I transfer all the solid into a recrystallization dish and manually removed all the remaining stearic acid. I place the recrystallization dish inside an oven and dry it about 2 hours at 100°C. At the end I got this nice looking powder, but it still contains stearic acid. I weighted the resulting powder, and in total I got 29.7 grams of not the purest PABA in the world. This corresponds to a yield of 74.29%, but considering the purity, it should be between 65 and 70%. To finish the experiment, I took the experimental melting point of the sample, and it resulted to be between 181 and 182 C, which is not very far from the theoretical value, but significantly below. For the aniline synthesis, let's start by weighing out 20 grams of para-aminobenzoic acid in a round bottom flask. I prepare the glycerin vat and immediately set up the simple distillation apparatus. As the vat reaches the melting point of para-aminobenzoic acid, some bubbles start forming. This is CO2. After some minutes, the distillate starts coming out. The mechanism goes like this. First, oxygen forms a double carbon-oxygen bond, and the electron pair between the benzene ring and the carbonyl group takes the remaining proton, forming aniline and carbon dioxide. I used aluminum foil to isolate the system. Eventually, all the para-aminobenzoic acid completely melted. After the distillation, this residue remained in the flask. And here you can see the distillate. There are some droplets of water, so we have to dry it out. I dried the aniline using approximately 2 grams of sodium hydroxide, pre-dried by heating it at approximately 100°C during 1 or 2 hours. After mixing very well the sodium hydroxide and the aniline, I filtered it through some filter paper, but here I committed a horrendous mistake. The filter paper had a small hole in the bottom, so I had to filter it again, losing approximately the half of the distillate I got. Very painful mistake. At the end I got 4.1 grams of aniline, which represents a very bad yield of 30%. Thank you for watching, please subscribe, and see you in the next video.